Welcome to part 2 of the lecture series dealing with an introduction to metabolism. In the previous lecture, we discussed multiple topics and introduced several concepts. We included a discussion of what metabolism was in general. We talked about the breakdown of metabolism into two broad categories known as catabolism and anabolism. We discussed that unique give and take relationship between catabolism and anabolism. We also spent some time talking about energy, and more specifically, free energy in a system. We then went on to discuss a little bit about free energy diagrams so that we could get an understanding of different types of chemical reactions that either required the input of energy or expelled or expended energy as a result of those reactions. We then rounded out the lecture talking about enzymes, and we began the discussion of what an enzyme really is. An enzyme is a catalytic molecule that speeds up a chemical reaction but does not get consumed in that reaction. What we also discussed at the tail end of the last lecture was how enzymes actually play a role in allowing reactions to be sped up. And we talked about a number of ways that enzymes can help contribute to the speeding up of a reaction. But now we're going to look at that in a little more detail, and we're going to draw on the concepts that we discussed with free energy diagrams. So before we do that, we need to define what's known as the activation energy. The activation energy, typically noted as the capital letter E with a subscript capital letter A, is the amount of energy reactants must absorb before a chemical reaction will start. So reactants must absorb energy from their surroundings to surpass a hill or a barrier, and that hill or barrier is known as the activation energy. So if you look at the very simplified form or version of a free energy diagram on the left-hand side, what you can see is the progress of the reaction along the x-axis and the energy associated with the components along the y. Again, in this very simple view, you see a certain amount of energy or free energy associated with the reactant in this reaction, and a lower level associated with the products of this reaction. So using those concepts that we discussed in the previous lecture, this is referred to as an exergonic reaction or an energetically favorable reaction. That is because the delta G is less than 0, and again, it defines an energetically favorable type of reaction. But what this very simplified diagram does is also include the activation energy, the energy barrier that needs to be overcome and absorbed by the reactants before the reaction can actually proceed. And that's denoted as that little wall or hill or barrier between the reactant and the products. So let's look at the right-hand figure. This one is a little bit more detail-oriented as far as a free energy diagram is concerned, but again, you read it the same way we've discussed previously. Progress of the reaction along the x-axis and the amount of free energy associated with the components along the y. So in this simple reaction, we have molecule AB and we have molecule CD, which are both reactants in this reaction. There's a certain amount of free energy associated with those two components. Then if you look at the far right of the figure, you can see the products of this reaction, which is now molecules CA and BD. There's been a breaking of chemical bonds and new chemical bonds forming when you look at the molecules that are considered the reactants versus those that are the products. But the important point here is that the free energy associated with the products is at a lower level than that of the reactants. This is also an exergonic reaction, an energetically favorable reaction, and the delta G is going to be less than 0 based off of the definition of how delta G is calculated, like we just reviewed. But what this figure also does is include the activation energy that is required in order for this reaction to actually proceed. The reactant molecules must absorb energy from the surroundings in order for those bonds to break. And very clearly in the center of this figure, you can see that activation energy, that hill or that barrier that needs to be overcome before the reaction will proceed. Now, let's look at the reaction once again and try and analyze or interpret what's going on. So in the figure that you see here on the left-hand side that is labeled B, again, we're looking at this very simplified version of a free energy diagram with the progress of the reaction along the x-axis and the free energy associated with the components along the y. Again, certain amount or level associated with the reactants, certain amount or level of free energy associated with the products, still an exergonic reaction. Delta G is still less than 0. But what you're seeing here in this diagram is that there is still an activation energy barrier that needs to be overcome. But if you compare that to the figure we looked at on the previous slide, that barrier is much lower. That barrier is now lower because of the help of the enzyme. So this is what this figure is trying to convey, is that when you have an enzyme that is present and able to assist a reaction, the job of the enzyme is to reduce that activation energy barrier so that the reaction can progress or proceed more readily. So now if you look at this more detail-oriented version of a free energy diagram, again, progress of the reaction along the x-axis, free energy associated with the components along the y-axis. We're looking at this view where you have a certain amount of free energy associated with the reactants, a certain amount of free energy associated with the products, and this is again an exergonic type of reaction with a delta G that is less than 0. The first that I want to mention is the curve in the black. The curve that is shown in the black on this particular figure shows the hill or the barrier that needs to be overcome, the activation energy that needs to be overcome before the reaction will proceed. And so you can see that also listed as the activation energy without the enzyme. However, what's superimposed on this figure is a curve that is in red, and that represents what happens with the help of an enzyme. As we discussed in the last slide, the enzyme helps lower the activation energy, lower that barrier so that the reaction can progress or proceed more readily. So in this case, what you're seeing is no change in delta G in this reaction. The delta G stays exactly the same. What changes with the help of an enzyme is the amount of activation energy has been lowered, and there is a distinct difference there between without an enzyme compared to with an enzyme. Most enzymes are proteins. Now, there are a few exceptions to that rule, and we'll talk about that later on in the semester. But for now, let's just think about enzymes, and the largest class of molecule that are in fact enzymes or have enzymatic activity are proteins. So what you need to do is go back and think about protein structure overall. In our previous unit, we discussed proteins. We talked about the primary, the secondary, tertiary, and quaternary levels associated with protein structure. One of the things that I highlighted at that point was that when you have a linkage of amino acids, one right after the other, that is a polymer that is made up of many amino acids, that's referred to as a polypeptide. Once you have that polypeptide, it begins to fold in its final functional three-dimensional shape. Then it can be considered an active protein. And it is the sequence at the amino acid level, the primary structure, the secondary structure with its alpha helices and beta pleated sheets, and the tertiary level that allows for the complex three-dimensional shape to form that again contributes to the overall function of the protein. That being said, proteins are extremely sensitive because of this three-dimensional structure. Many different components, physical and chemical, can play a role in altering that three-dimensional shape. And when that three-dimensional shape is altered, the function of the enzyme is also altered. So first, let's focus on temperature. Again, you have a protein that has a very complex three-dimensional shape. And if you alter the temperature that you find that enzyme in, that could play a really big role in denaturing or causing that protein to unfold. If the protein unfolds, it's no longer in its final functional conformation. So in the top half of this graph, what you're looking at first is optimal temperature for two different sets of enzymes. Along the x-axis, you have a variety of temperature ranges from 0 to 100 degrees Celsius. And then you have the rate of the reaction along the y-axis. Now, in this example, what you're looking at in the first curve is the optimal temperature for most human enzymes, and that happens to be right around 37-38 degrees. This is the body temperature for humans, and it makes sense that most enzymes that are found in the human body operate at that optimal temperature. If you look at the graph that's shifted from that with the green curve, you're looking at the optimal temperature for some thermophilic bacterial enzymes. So there are bacteria that live in thermal hot springs where the water is near boiling. You can imagine that probably wreaks havoc on enzymes because boiling proteins tends to denature them. So evolutionarily, those organisms that live in these very extreme environments also have enzymes that have evolved in a way to be able to remain functional at these extreme environmental conditions, such as temperature, like you see here. If you took one of our human enzymes, where the optimum is right around 37-38 degrees, and you shifted that to almost boiling point, the protein would denature and the enzyme would no longer function. Perhaps the converse is true as well. If you took one of these enzymes from a thermophilic bacterial species and put it at a very low temperature, the enzyme most likely will not function. And based off of the graph here, it shows that it would not, in fact, function. So again, enzymes are extremely sensitive, and they evolve to function at their optimum, typically in the environment that you would normally find that enzyme in. So if you focus now on the bottom portion of this figure, another property that can affect enzymatic activity is pH. In this figure, again, you're looking at a range of pHs from 0 to 10 in this case, and the rate of the reaction of whatever reaction it is we're interested in along the y-axis. Here too, you have two different sets of enzymes. In this case, however, they are both enzymes that are isolated from humans. They are digestive enzymes. However, one enzyme that is shown with the black curve, known as pepsin, is found in the stomach. And what do we know about the pH of the stomach? Well, the pH of the stomach is extremely acidic. So an enzyme that's found in the stomach needs to be able to withstand the pH found in the stomach to remain active and functional. On the far right then of that graph, with the green curve, you see another digestive enzyme known as trypsin, and trypsin is found in the intestine. In this particular case, the pH is no longer acidic, and its optimal function is at a pH of close to 8. And so, again, you have two different enzymes now from the same species, humans. However, where they're located definitely plays a role in their optimum activity overall. If you took pepsin and treated it with a pH of about 7.5, you would see that enzyme would no longer be functional. And conversely, if you took trypsin and put it into an acidic solution of about a pH of 2, again, you would see that the enzyme would not function. So these enzymes have evolved to function at their optimum in the environment that you normally find them in. Now what I want to do is expand on those concepts of physical and chemical properties that affect enzyme activity. What I want to talk a little bit about are two categories of molecules that can interact with enzymes that play a role in their function. The first category is a molecule known as an activator. An activator does exactly what its name implies. It is a chemical that must bind for the enzyme to be active. It plays a role in allowing for the enzyme to be active. There are two broad categories of activators. There are cofactors, which are non-protein molecules required for enzyme function, and then coenzymes, which are protein-based molecules required for enzymatic function. So at this point, I usually like to deviate just a little bit and ask the class, does anybody take a multivitamin? And oftentimes, most of the class raises their hand and say, yes, they do. If you looked at your multivitamin ingredients, you would see that not only are vitamins included, but many also include minerals, things like magnesium, zinc, manganese. Those are all components that are considered cofactors and that they are essential for enzymatic activity. So when you take a multivitamin, you're not only supplementing your body with vitamins, but you're also supplementing your enzymes with the activators or cofactors that are necessary for your enzymes to function at their optimum. So converse to activators, you have inhibitors. And inhibitors also do exactly what their name implies. They are chemicals that turn off or shut off enzymatic activity. There are two broad categories of inhibitors. There are those that are considered competitive inhibitors. These are structurally similar to the substrate overall and tend to back or block the active site and hence are competing for the active site. If they bind or block the active site, then the true substrate cannot bind. So in the figure that you're looking at on this slide, on the far left, you see the classic interaction between substrate and enzyme in the purple and substrate in the blue. With a competitive inhibitor, you have a molecule that is, again, structurally similar to the substrate and can bind to the active site and block the active site, preventing the true substrate from binding. And in this case, you have an inhibition event taking place. A non-competitive inhibitor is like what you see on the far right. A non-competitive inhibitor binds to a site that is other than the active site. This is referred to as an allosteric site. When it binds to the enzyme in this allosteric site, it causes a conformation change in the enzyme, making the active site not ready or available or the right shape to accept the substrate, and hence prevents the reaction from taking place. Now we're going to transition just a little bit in topic. However, it still does tie in with our overall intro to metabolism, and that is to talk about the concept of ATP and cellular work. There are many types of cellular work that needs to occur. That is, jobs that need to take place in the cell that require an input of energy. They typically fall into three broad categories. Things such as mechanical work, that is movement overall inside the cell or the cell itself. Transport, that is moving molecules across membranes. And then chemical work, which is allowing for chemical reactions to take place. So on the right-hand side of this figure, you're looking at a figure that we looked at in our previous lecture, and I just want to recap what we're looking at. This is a summary overall of metabolic reactions and how there's a nice give and take between the two broad categories of metabolic reactions. If you recall, catabolic pathways or catabolism involves the breakdown of large molecules, typically macromolecules that are some food or fuel source, breaking them down into much smaller components that can be utilized in the cell. However, when these catabolic reactions occur, there is in fact energy that is released from those large molecules that can then be shunted or shifted over to the other reactions, which are known as anabolic reactions or pathways. Those reactions take small molecules and build up larger molecules and do require an input of energy. So again, nice balance or give and take between energy being released from catabolic reactions and energy needed for anabolic reactions. So this then leads us to how does that shuttling of energy from the catabolic reactions occur to the anabolic reactions? And this occurs with the help of a general molecule that is categorized as an activated carrier. The job of an activated carrier is to carry energy or chemical group in many different types of metabolic reactions from one reaction to the other. They're basically acting as a shuttle or a transport mechanism to transfer the energy from one set of reactions to another. And the main type of molecule that does that is a molecule known as ATP. ATP is short for adenosine triphosphate, and in this very simple figure that you see on the left-hand side gives you an idea of what's happening with that shuttling of energy. And we're going to look at it in more detail in the next slide. But what you see here is energy that is being released from catabolic reactions is incorporated into a molecule known as ATP. And it's always shown with these little lightning bolts or flashes of light associated with it to indicate that it has a great deal of energy associated with it. So energy is transferred from the catabolic reactions where it's being released to this form of energy in the molecule known as ATP. ATP can then transfer that energy, act as an activated carrier to those reactions that need it, like anabolic reactions. And when that happens, ATP is converted into ADP and an inorganic phosphate is released. And again, we're going to look at that in more detail on the next slide. So what you see here is a very simplified view of ATP, which again is short for adenosine triphosphate. If you look at the figure, what that shows is there is an adenosine molecule, shown very simply as the blue box, and 3 phosphate groups that are linked one right after the other covalently. ATP, the triphosphate molecule, contains a great deal of energy. What it does is transfers that energy to the reactions that require it. When it does so, it is converted into ADP, which is adenosine diphosphate, and an inorganic phosphate is released. Oftentimes, it's transferred to the molecule in question during the transfer of energy reaction. Now, this figure shows a little more detailed view of the molecular structure of ADP. So adenosine is actually the molecule adenine and the sugar ribose, and 3 phosphate groups linked one right after the other. So adenosine triphosphate is the molecule that you see in panel A, known as ATP. What I want to highlight with this figure is the fact that there are 3 phosphate groups covalently linked one right after the other. And essentially what you have here is a series of negative charges because of those phosphate groups having negative charges one right after the other. And this is in a sense like a loaded spring. If you take a spring and scrunch it down, it has a great deal of energy associated with it because as soon as you release the spring, it's going to pop up. By having those 3 phosphate groups covalently linked, we've now essentially squished down the spring and made it so that there's a great deal of energy associated with it. This is an extremely unstable molecule, and to make it more stable, it would be advantageous to release some of that tension, basically releasing one of the phosphate groups. In panel B, what you can see is the reaction that takes place when the energy is released from ATP. It is a hydrolysis type of reaction, and an inorganic phosphate is released in the process overall. So let's focus in a little more detail mechanistically what is happening with the transfer of energy from ATP in these reactions. So we talked about the 3 broad categories of work. We have mechanical work, we have transport, and we have chemical reactions. The mechanism of action for ATP providing energy to those reactions is similar in all examples, but we're going to go through each one of those now. In panel A, we're looking at mechanical work. So this might be a situation where you have a motor protein, again, like we talked about when we discussed the cytoskeleton, and movement taking place. It does require the input of ATP or energy to allow that movement to occur. So the energy associated with ATP gets transferred to the motor molecule in the form of the phosphate group from ATP being transferred to the motor molecule. This then makes ATP ADP and much more stable. However, now the motor molecule has the phosphate group associated with it, making it extremely unstable or reactive. This then is what allows for the actual movement to take place. Once the phosphate group is released, there is much more stable state, and again, the reaction can continue or repeat over again. The same mechanism is occurring in panel B. In this case, we're talking about transport with a transport protein. Energy is required for transport proteins as part of the active transport mechanism. We discussed that ATP produces or provides the energy for those reactions. The mechanism, though, again, is the same. The phosphate group from ATP gets transferred to the transport protein. That then causes the transport protein to be unstable, typically causing a conformation change, which usually allows for the molecule in question to pass through. Once the phosphate group is removed, the transport protein resumes or becomes the original shape, and the transport has occurred across the membrane. The end result, though, is that phosphate group being released and the energy from ATP also being released to form ADP. Lastly, chemical reactions do in fact require an input of energy. Many of them do. And so in this particular case, the mechanism of action is still the same. The phosphate group from ATP is transferred to one of the reactants, making that reactant now unstable, allowing for the reaction to proceed. Once the phosphate group is removed, the reaction is complete, and you are left with ADP and an inorganic phosphate. So in the previous set of slides, we discussed how ATP has a great deal of energy associated with it, and mechanistically, how is that energy being transferred? It's through the transfer of the phosphate group that then makes the molecule in question reactive or unstable to allow the reaction to proceed. But now what we're going to do is we're going to superimpose that with our free energy diagram. So in this free energy diagram, this is a much more complex free energy diagram. You still read it the exact same way we've been doing. Progress of the reaction on the x-axis, the amount of free energy associated with the components along the y-axis. Let's focus first on the chemical reaction here. We have reactants A and B, and this is going to be a synthesis type of reaction where the individual unit B is going to be linked on to the chain represented by the letter A. If you look at this reaction, the amount of free energy associated with the reactants is at one level, and over on the right, the amount of free energy associated with the products is at a higher level. This is an endergonic type of reaction. The delta G is going to be greater than 0, and in this case, what we need to do is we need to input energy into the reaction. How is that going to happen? We're going to couple that with the reaction that's superimposed on this figure, and that is of ATP going to ADP. The amount of free energy associated with ATP is very high, whereas the amount of energy associated with ADP is much lower. And so the energy that's released from ATP going to ADP is going to be coupled to this reaction that we just talked about, where the individual unit B is going to be added on to the growing chain A. And again, this is how the reaction will proceed, is energy from the release of one reaction is going to help fuel or allow for the other reaction to proceed. So one more concept that we're going to end this particular lecture series with is talking about another set of reactions that occur in the cell. These reactions are known as redox reactions, and that is short for oxidation reduction. So the RED from reduction and the OX from oxidation forms the word redox, and this represents a class of reactions. In a redox reaction, you have a chemical reaction involving the transfer of one or more electrons from one reactant to another. This also is an example of a transfer of energy because electrons have a great deal of energy associated with them. So in the figure, you have many examples of the redox reaction. Let's focus on the first one that's in the center of the slide. You have reactants A and B, and in this redox reaction, what is going to happen is there's going to be a transfer of an electron from reactant A to reactant B. In this case, what you have is the oxidation of A and the reduction of B. The end result is that you still have A and B, but now A has been reduced by an electron. It's going to have one less electron associated with it, whereas B is now going to have an additional electron associated with it. In the bottom two figures, you are looking at the same concept. Again, generically on the left-hand side, you have X and Y. In this case, reactant X has the electron that is going to be transferred to reactant Y. X is undergoing the oxidation. Y is undergoing the reduction. And I also oftentimes like to add why these terms are the way they are. When you talk about the reduction, what's happening is you're reducing the amount of positive charge associated with that component. Another classic example of a redox reaction is the formation of sodium chloride. We talked about sodium chloride when we talked about ionic bonds forming, but this is also a type of redox reaction. The electron associated with sodium is going to be transferred to chlorine, hence sodium is going to undergo an oxidation, chlorine is going to undergo a reduction. Now, ATP is not the only activated carrier in the cell. Another common activated carrier is a molecule known as NAD, nicotinamide adenine dinucleotide. This is also an activated carrier in that NAD is a molecule that can pick up electrons, and in that case picks up electrons and undergoes a reduction. So NAD becomes NADH. NAD does not have a great deal of energy associated with it, but NADH does because it now has those electrons associated with the molecule. This is going to be really important as we transition into Unit 3, and so you're going to have to recall this information as we move into Unit 3. But for now, we'll just leave it as NAD being another form of an activated carrier. In this case, picking up electrons, and electrons also, again, have a great deal of energy associated with them.